No one actually said anything.
Thymine.
Right. So if this was DNA with deoxyribose here, because of the structure, instead of uracil, you would have thymine, and it would be a T there. But no Mr. T here. We pity the fool who draws a Mr. T in RNA. And then you're going to have the 5 end here, that's the top, and 3 end there, the 5 end there, 3 there, 5, 3. So this is an upside down RNA chain. This is right side up, goes bottom to top. Way to remember this, if it ever comes up, is low to high, low to high, low to high. And so you'll have your 3 going upwards to the 5. 5's bigger than 3. Same with this going upside down. And one way, we're going to zoom out even more.
Like, you'll receive a question that says, which way does DNA replication occur? 5 to 3, 3 to 5?
Yeah, we have seen that a couple times.
And that comes up like almost too often to miss, you know?
Yeah. So you're going to go up and G, C, U, and RNA is tricky. It's a single strand, but it likes to bend around because it has the freedom to do so. So you'll have some bits that will want to grab together, but you'll have other bits that will just make a really funky loop. Like, you don't see that in DNA. DNA is simple. You have 2 strands like that, and then it coils.
So RNA viruses are coming.
Could I interrupt?
You can interrupt.
Could you do it with a darker marker?
Nah.
This? Yeah.
Thank you.
I'll do purple.
This is why RNA viruses are also more scary than
Right.
viruses.
So yeah, like Osman said, RNA can be a lot more scary. Or RNA-based viruses can be a lot more scary. Oh yeah, stating the obvious, because I don't think I have yet, DNA and RNA code for genes, and they're important because they define how like your body will make things later. These code for proteins, something I'll get to later. Just wanted to say that in case someone in here didn't know that, or someone watching this video didn't have that assumption coming into this. Yeah, it's something I should have said at the beginning. So you'll get these pairs, and these loop up, these loop up, these loop up, but these don't. Uracil and guanine don't like each other. They don't bind, because uracil will have 2 hydrogen bonding pairs that it can loop up with, and guanine has 3. And so they won't bind, and you'll go around, and it'll just be just some loop. And because of this, there are weird shapes. When you actually wind this up, because with the double helix, the way the structures work with the phosphates and the ribose binding, it tends to twist. Same thing with deoxyribose. So you'll get something more like this, where the twist is more pronounced. And instead of smooth DNA, like that, it tends to bend more to one side. So you'll get a thicker curve. And at the top, some of these will actually stick out at times instead of being in. So if you're just looking at a picture and you don't know, like, and you can't think, well, is that DNA or RNA? 3 ways to find out. 1, if you're told if there's uracil in it, it's RNA, and if there's thymine, it's what? DNA. 2, there's a loop. There's a really, really big loop at the top, because RNA is 1 strand, and it will loop back on itself and make these segments. 3rd is what?
The curve.
Yeah, the thicker curve. It'll bend more to one side and the other, whereas DNA is always symmetrical. It'll balance out on either side. So if you were to have a top view of these, you would get something that looks like, where's my green marker? Here it is. You'll get something that looks sort of like this, and there's a hole in the middle. Whereas DNA, because it's perfectly symmetrical, how do you think these lines will cross? Right, always through the middle. So it looks like a bunch of X's like that. So these go straight across. But RNA, if you're looking from the top, there's a hole in there because it'll lean to sort of the side of what you consider the axis of the whole thing. So we've covered almost everything possible to cover about naming the bases and the actual structure. So in just a second, we're going to go into how this actually works, like what will happen when you actually use DNA and RNA together to start making proteins. Does anyone have any questions about anything we've covered?
If you don't have any questions, you're lying.
Yeah. Anyone? Anything confusing? Anything you want me to go more in depth on? Okay, I'm going to ask you some questions just to review. So what is the difference between ribose and deoxyribose? In the back.
Deoxyribose has oxygen taken away from it.
Right, deoxyribose has oxygen taken away from it on the carbon 2 atom, the second one in the ring. And what does this 1, 2, 3, 4, 5 mean?
The order of the carbons.
Right, the order of the carbons, just starting from the nitrogenous base because that's the most important thing in this. If you have a more normal sugar that has 6 carbons, you would number it 1 through 6, and it would work like that. Since this has 5 carbons, what do we call this kind of sugar? Pentose, right. And if it were 7, it would be heptose. If it were, it doesn't go to 4. You don't have rings of 4. But then you just have the prefix. And so all of these names come from just the simple ideas that, like, each little bit describes something else about it. So if I had, say, deoxythymidine triphosphate, or DTTP, or let me write that in dark colors for the video. DTTP, that didn't help.
Third time's a charm.
DTTP.
Erasing still exists.
What is inside this? What makes up DTTP? Say that louder. Right. Right. Well, it's deoxythymidine triphosphate. Right. So deoxythymidine means that it has deoxyribose tied to it. It's dene, so it's a pyrimidine, and it has thymine. The triphosphate means 3 phosphates, obviously. And with the way they're written, you'll have like ATP if it's attached to a ribose, or DATP if it's attached to a deoxyribose. Osmond, anything else from this section I should cover?
It seems to be pretty comfortable. What about nucleotides and nucleosides?
Oh, yeah. What's the difference between a nucleotide and nucleoside? What's the one I just drew up there with a triphosphate? Is that a nucleotide or a side? How many parts to it does it have?
The nucleotide.
Nucleotide. So DTTP has deoxyribose. It has the thymine group and it has 3 phosphates. So 1, 2, 3, and you're going to have nucleotide, the T for 3 parts. That's how I personally remember it. So just a second. You have the sugar, and you have the nitrogenous base and the phosphates, however many there are. And if there were 2 bits, if there weren't phosphates, what would you call it? Nucleoside, right. You're thinking if I only had 2 phosphates, so I'm saying no phosphates.
Oh no.
Then it would be just,
They also go in
Thymidine.
alphabetical order. So, they're great, like, you know, DST, like the tide, die, side.
oh, hey, they do. So yes, you had a question.
What's the difference between side and tide again?
Okay, so that tells you if it's a purine or a pyrimidine. So you remember you had your, what was it, the adenine and guanine. Their basic structure was like a hexagon and a pentagon bound together. Whereas uracil, thymine, and cytosine had just one hexagonal ring. And so this will have, when it goes into a nucleoside with 2 parts, no phosphate, this will have the suffix sine, and this, which cytosine already has that S, will go into dine. So you'll have adenosine or uridine, and so on. So now that we've covered all of this, I'm going to go into transcription and then translation with